16th season of Bringing You to You here in Great Falls. Let's have a hand for each of you. All the people that are here. Thank you for coming. You know, we have a committee that works this, and we always are apprehensive about knowing if we got topics that people would like to hear and whether we're doing our part of the job correctly. Bill Betcher, where are you? Bill, would you stand up, Bill? Oh, he's out there. Bill, come in the door, Bill, would you please? And all the members of the Bringing You to You committee, would you please stand? Okay. Thank you, guys. You know, we serve with mixed reasons whenever we're doing something with the alumni groups or with our alma maters, but the payback is having people come, and we're really glad to have you here. I'd like to thank, we'd like to thank Montana State University of Great Falls and for all the good things they do for us here. The amount of support we get from the IT people and from the people who set up the rooms, and wherever that cook is that cooks those cookies. They do a really good job. Thank you for that. How many of you are here are teachers? Would you raise your hand if you're a teacher? Yeah, okay. Ann Simpkins is right here. Ann, okay, so if you're looking for Ann afterwards, she needs to sign you up. We're terrific that we're glad that you're here. Finally, Montana State has a university table over there with free giveaways. Feel free to take anything that's on there. The university table will be here next time, and we'll have that there for you also. We have a representative of the Montana State Alumni Foundation, Michaela Bader. Michaela, there you are. Thank you for coming up, Michaela. You got any questions or you'd like to discuss further with her or anything about it, go run her down. Okay, you didn't come here to listen to me, but you're going to have to listen to me for one more minute. I'm a chemical engineer who graduated from Montana State in 1963. And I think you have to recognize that things have really changed. Maybe in chemical engineering, some of the things that have changed. We had 28 graduates, and we were the largest class that had graduated in Chem-E when I graduated in '63. And 10 of those students were foreign students, so there were 18, mostly Montanans, who graduated then. Tonight's speaker is a chemical engineer, and in his resume, if you saw where he's been, he's got his undergraduate in Florida. He then went to work and ended up coming to graduate school at Montana State in the mid-'80s. And in his resume it said he went there to further his work in chemical engineering. And the chemical engineering instructor, Trackless was his last name, got him interested in biology and the chemistry of biology, which for most of us chemical engineers didn't have biology after high school. That was the last time I had any biology. So he'll probably tell you the rest of the story, but suffice it to say, we've come a long way, both at the Montana State University in chemical engineering and in the breadth of what we're doing chemical engineering-wise. And it starts with a thing called research, and researching these new ideas that are taking us into new areas. And I'm not going to get into it anymore. Brent Payton, why don't you come on up, Brent? Brent has been a lecturer in the Provost series down in Bozeman. We saw that, we liked it, and so we've invited him up, and I think you're going to really like him. Can I introduce your wife? Sure. Yeah, and brought his wife Kathy with her. Kathy's a Butte girl, Okay. So, we have ties in a lot of different places. But, go ahead, Frank. I'll turn it right over.
Can you guys hear me? This feels a little Britney Spears-ish, but seems to be about the best microphone. Thank you guys so much. I want to thank the organizing committee for inviting me. I want to thank everybody for coming out tonight, and hopefully this will be an interesting presentation. These are very different for research professors, faculty, because the provost asked us to lead a little bit of, give a little bit more background as to how we ended up doing the research that we do at Montana State University. So got a little bit of history, a little bit of my history, a little bit of all the research areas that I've worked on. I'm not going to go into anything in too much depth, but hopefully you enjoy it. So I'm going to start with my parents. They did a great job raising me, I think. My dad was a chemist in Texas, North Texas State University, and I always wanted to do what he did, but thought of myself as a little more practical, so I did chemical engineering instead of chemistry. And so even in 11th grade, I got into a program up at the University of Georgia, doing research for the summer, and got a job at a consulting firm after that through a 12th grade community science leadership program. And it was actually my chemistry teacher in high school that made me decide to be a chemical engineer. She's like, you are good at math, good at chemistry. She's not as scary as she looked in the Usually not. She was a great teacher, very stern. So after high school, I went to University of Florida. I grew up in Gainesville, after Texas, Gainesville, Florida, and went to the University of Florida, started my freshman year, but I didn't really do so good in my freshman year. In my second semester, I kind of started skipping classes and not going to class and stuff, and so I ended up moving out to Texas to live with my dad and took up canoeing for about a year. And we went all over the place, did a lot of canoeing. But I knew in the end I wanted to go back to school, so I went back to University of Florida and pretty soon became the president of a canoe and rafting club. Chemical engineering and canoeing and rafting, and led a bunch of trips up into North Carolina and Georgia. This is the Chautauqua River where they filmed Deliverance. Anybody's ever seen Deliverance? And so anyway, had a good time doing a lot of outdoor kind of things, and then also working on my degree. So, Trailways, how did I end up in Montana? Trailways had a round-trip bus ticket to anywhere for $69. And I thought, what's the most exciting place I could possibly go? And it turned out that it was Great Falls. It's an 80-hour bus ride to Great Falls. I got off here at the station and hitchhiked to Augusta and went up into the Bob Marshall for about a week. Turns out in Florida it's quite warm in June. In Bob Marshall, this is about June 1st, and not very warm, so it's very interesting to be making snowmen in June to a kid from Florida. Ended up hitchhiking around the state for about a month, maybe 6 weeks, and then decided that I was going to go to grad school up in Montana. So basically that's how I ended up here. I finally graduated. My grandpa gave me a 73 Gremlin. And with my 2 canoes and a mountain bike on the roof, drove up here, got to Bozeman, came over Bozeman Pass January 2nd in a blizzard. Anyway, I thought I was going to die like 20 miles from Bozeman. So I ended up joining a research group. This is Bill Shiraklis. He was the head of a small group called the Institute for Process Analysis. And very cutting-edge research. And it turns out just about all the students in this group ended up being professors. Bill was such an inspiring mentor that everybody wanted to do what he did. So we studied biofilms. These are the organisms that grow on surfaces. Well, the worst example is on your teeth, right? Inside your toilet bowls, inside pipelines and industrial systems. And so actually most of my PhD work was funded by Conoco to try to figure out, they had big water injection lines that had biofilms that were plugging up the oil fields that they wanted to try to eliminate these biofilms. So these are cells growing on a surface. They can detach and attach, and basically as a chemical engineer we put mathematics to these things and try to figure out how to control them. Also did a lot of work with a guy, his name's Dave Ward. He liked to work hard, but he never finished with the play hard. How it's supposed to be balanced. But he taught me all about, took us to Yellowstone in some of our classes, taught us all about organisms that live in very extreme environments. So if you think about what's kind of normal, like rivers in summer that you would swim in, but then there's organisms that live in very, very different conditions. So things like thermophiles in Yellowstone live at very high temperatures. So if you go home tonight, turn your hot water tap on, most people's hot water comes out at about 60 degrees centigrade. Very hot. There's some organisms that just can't even live in that cold of water. Same with organisms that live at very cold temperatures, some that live in acids, some that live in very strong bases. And so there's a whole range of organisms out there that people hadn't really studied much before. So Bill was an amazing guy. You never quite knew what was going to happen. He was very personable, charming. About the same time, I had met my wife, and we ended up getting married in '91. So that was I traded that Gremlin in for a 65 Ford. I think that was much nicer from Montana. Unfortunately, Bill passed away in 1992, about a week after I graduated. And so this was his favorite saying. I've got a copy of that on my wall also. But, amazing person. I don't know what things would be like if he had lived to be 70 to 80. So after I graduated, we ended up getting a job at the Pacific Northwest National Laboratory. It's a Department of Energy laboratory out in Washington State, where they built did a lot of the reactors for making the bombs used in World War II. So a lot of nuclear waste out there. The area that I worked in was a pretty industrial-looking place. And the building that I worked in was right over here, right near where they stored the plutonium. So my first job, coming straight from Bozeman, to get into my office, you stepped into this little cage and held your badge up to a camera. If that looked okay, they let you go to the guardhouse where they had the guards with the metal detectors and the automatic Uzis. And if you still looked okay, then they'd let you go to work. And so worked there for about 5 years. Started this working on a project called bioremediation. So it's using microorganisms and understanding of engineering to clean up contaminants that were around in the world. And one of the biggest ones that we worked on was this jet fuel spills at Air Force bases. So I spent a lot of time at Tinker Air Force Base in Utah, a lot of time, or in Oklahoma, thank you. The other one's in Utah. Hill Air Force Base in Utah. And basically the idea behind this was these organisms were down living in this contaminated jet fuel underground. If you blow enough air in there, all they needed was some oxygen, and they could break down the jet fuel and turn it into carbon dioxide and decontaminate the soils. And so this has been a very, very successful technology, and we're part of the group that developed this and documented that it was working. Some of the jet fuel sites that we worked on had 6 feet of jet fuel floating on the groundwater surface underground. So we would pump all that jet fuel off and then bioremediate what was left. The other area that we worked on, carbon tetrachloride, was used at the Hanford site to help purify plutonium. They would put it in a a disposal trench every afternoon, I guess, and by morning the trench was empty, and so they could put more in. And so eventually it ended up getting down into the groundwater, and at the time that I was working there, Hanford had a 7 square mile plume of carbon tetrachloride. I don't know what it is now. But we developed a microbial process that could break that carbon tetrachloride down, and the base would be contaminated. We ended up injecting acetate, which is basically vinegar, into 200 feet underground, feeding the natural microorganisms that were there, and that they could break down this carbon tetrachloride so it wouldn't continue to move. The process looked like this. It turns out we remediated probably an area, maybe 30 meters long and 20 meters wide, compared to a 7 square mile plume. That was nothing. It turned out it was just going to be too expensive to remediate the Hanford. But this technology's been used to clean up a lot of chlorinated solvents at other Air Force bases, at dry cleaner places, things like that. So it's still very useful, but not on a plume of that scale. So we started looking for other things to work on that we could use microbes and engineering combined to clean up a variety of contaminants. And one of the ones that we looked at was cleaning up heavy metals like lead, copper, cadmium, nickel, and zinc. There's organisms called sulfate-reducing bacteria that can take sulfate and convert it into hydrogen sulfide. The sulfide reacts with the metals and precipitates them out. Many of these down below drinking water standards. And so you can grow these organisms, they're everywhere. We could go out and get some mud in the river and do this process and decontaminate these metals. About that time, we started working on another group of organisms called metal-reducing bacteria, and they could take uranium, chromium, technetium, and selenium and precipitate those as solids so that you can at least keep your contaminants inside the borders of what you know is contaminated so they wouldn't spread. So we could feed them things, again, like vinegar, sugar, a variety of just very common things that people would eat and precipitate these metals. And I'll get back to the selenium one and uranium actually. So this is what some of the laboratory data might look like. We would put in these red lines are uranium concentrations in the water. And so we would add the bacteria, give them the food, their food is blue, and they would start to grow, and the uranium concentrations would drop down to drinking water standards in about, maybe a month. And so we could use this process to clean up uranium out of contaminated water system, groundwater systems. We also started looking, Hanford has these large tanks, millions of gallons of radioactive waste that are very high pH, very high salt. So we started looking for organisms that can live under those conditions. Using some of the information I learned at MSU on these organisms that live in extreme environments, we started looking for organisms that could live under these and do processing that might be beneficial for the tanks. And so we went to places like the Bonneville Salt Flats and found where the salt-soil interface, there'd be microorganisms living right there that could tolerate very high salt concentrations, very high pHs, and brought these organisms back into the lab and showed that they could, in fact, degrade nitrate, convert it back into nitrogen gas, which was non-toxic, and essentially leave the solution. Also, other places that have very high pH, like this is Soap Lake in Washington. I don't know if you've ever been through there. It's a very interesting area. And Mono Lake in California. Got organisms from there that could also do a similar process. So after 5 years at the Pacific Northwest National Lab, I had wanted to get back to the university system. Washington State University offered me a job. And so I had actually taught some night classes at a branch campus to make sure that I really wanted to teach, right? I hadn't done any teaching for many years, and I wanted to make sure I liked it. And I spent we spent 8 years there. And I'm not going to go through all the stuff, basically all the research that I did, but very similar kind of research, 8 years worth of it. And Pullman's a fabulous place to live, but when I was offered a chance to move back to Bozeman, we jumped on it. So I did a sabbatical in Bozeman in Gil Deasy's lab, and wanted to start looking at organisms from high temperatures. So this is the Heart Lake Geyser Basin in Yellowstone. That's Heart Lake back there. Standing here about 5.5 miles from the highway. So I wanted to kind of get back doing more outdoor activities and mixing that with some of the research interests. So got some good hiking and good camping and sampling these hot springs to see what organisms live back there. And it's a very, very diverse place. There's different springs that are close to boiling. This is called Witch Creek. It's pH 10 and about 55 degrees C, so in the 160 degree range Fahrenheit. Some of these springs we've never even sampled because they're too dangerous to even try to get close to. We're worried about collapsing and falling in. But a very, very interesting place to work. So while I was on sabbatical, I interviewed and then joined MSU in 2005, and I've been there since. So as soon as I joined MSU, I joined the Thermal Biology Institute, which is a group of faculty that study the microorganisms that live in Yellowstone, and essentially trying to correlate their biological activity with some of the physical and chemical processes, and really focused on organisms in hot springs in Yellowstone. So I want to give a little bit of background here on Yellowstone and some of that. It's most people know it's one of the largest volcano systems in the world. Here's a map showing some of the magma chamber as best as people know. Underneath Montana, here's Bozeman, so we're up here today. And it's basically, my understanding from reading is that the amount of magma underneath there is about the size or volume of 11 Grand Canyons. So if you took the Grand Canyon and filled it up 11 times, that much magma is under there. Okay. So let's talk about eruptions. I was talking about Mount St. Helens a minute ago. Mount St. Helens, if you take all the material that came out of Mount St. Helens in 1980, it's that little box right there. The amount of material that came out of the largest eruption from Yellowstone was 600 cubic miles versus 0.24 cubic miles of material. 600 cubic miles, it's hard to put that in perspective. And so one of the ways that I try to do that is just give you an idea. If you drove from Great Falls to Seattle, not a perfectly straight shot, but once you get on 90, it's not too bad. 640 miles. If you had magma 50 miles on each side of you, 50 miles out on each side, it'd be almost 50 feet deep. From here to Seattle, that's the amount of material that came out. So it's huge, huge eruptions. Or 21 feet deep on the entire state of Montana. It's a lot of material. Everybody would be living on the second floor. Maybe the third floor. So what's left? We have a caldera now in Yellowstone that's about 40 miles long and about 30 miles north and south. We have 11,000 thermal features that are about more than 50% of the world's thermal features in Yellowstone. Less than 2% of these have ever been characterized to see what microorganisms live in these. So we've got a lot of information on what's all these different thermal areas here, but only 2% has ever even been sampled to see what microorganisms might be living in. And Yellowstone's changed a lot, right? So it used to be bears begging for peanut butter sandwiches at the gates. And now it's radio collars and as natural as possible. It's kind of the same thing with the hot springs. It used to be kind of more gee whiz and But now we're starting to understand that each of these hot springs has unique environments in it, and the organisms that live here in this one are maybe drastically different than the organisms that live in the one next to it, and very different from the photosynthetic organisms that live in the steam on the edges of these springs. And so there may be 1,000 species here and a different 1,000 species there that nobody really knows much about. So that's kind of what we're trying to figure out. There's cyanobacteria, there's fungi, there's bacteria, there's viruses that infect those. Very complex group of organisms. These are called thermophiles, and the organisms that thrive at extremely high temperatures. So again, go home tonight, turn your hot water tap on. That's about 60 degrees C, so sort of right in the middle of these moderate thermophiles. There's some that require temperatures 80, 90 degrees centigrade, right up to boiling, and can live even above the boiling point of water. So boiling them doesn't kill them for these guys. They can also tolerate lots of toxic metals and high pH, so they can live in boiling acid, they can live in boiling alkaline solutions. Very, very tough little critters. So why study these systems? I mean, maybe you're kind of wondering, why would we even look at these? And, get an idea of the other biodiversity on Earth. I mean, we've got a lot of genetic diversity and potential solutions to future problems in organisms that we've never even seen or grown before. Provide clues to conditions on early Earth and evolution. So there's a lot of times believed that these thermophiles were some of the earliest known forms of life, organized forms of life. Only about 1% of these have ever been grown in a lab, so, and that's true for most bacteria. So we've got all the biotechnology, everything you read about biotech is based on about 1% of the bacteria that exist. And 99% are still out there waiting for us to discover and characterize. So lots of potential. Again, tremendous biotech potential. So, has anybody ever seen CSI? Where they find a hair from the criminal and they take it back to the lab and they amplify and sequence the DNA. That is made possible by an enzyme from an organism called Thermus aquaticus that was discovered in Mushroom Spring in Yellowstone Park in 1985. So it's what made CSI possible. The human Genome Project. So all of us learning about our genes and whatnot is made possible by this enzyme from Yellowstone. It's basically a DNA photocopier. You take one copy, make 2 to 4, 4 to 8, and finally you get enough DNA from that sample that you can sequence it and figure out who the criminal was. It's used in criminal identification. A lot of medical diagnoses now are coming out. The Human Genome Project that will yield many, many solutions to medical problems. And it's even used currently to manage Yellowstone's grizzly bears. When a grizzly bear commits a grizzly bear crime, they can get the hair and make sure that they've caught the right bear to dispose of or transport up to Bob Marshall. And they use DNA and enzymes from Thermus aquaticus to do that. So I got started looking at this, trying to find organisms that could break down organics at high temperatures and high pHs. We started with a small project from the Department of Defense looking at accelerated degradation of explosives at high pH. We accelerated the degradation, but not quite fast enough. They still like to light it. It's a lot faster than the bacteria. We're currently looking at ways to break down lignin and cellulose into renewable products, so using enzymes at high temperatures to accelerate the rates and getting something useful out of these, breaking cellulose down to sugars, and sugars can be fermented into ethanol, things like that. We haven't really done any work with pesticides or the alkaline wastewater, but it's something that I believe is possible. We just haven't had the time to get into it. Working in Yellowstone is very unique. I can't believe I get paid to do that. No shortage of volunteers to go help with the sampling. Over the years, we've done a number of horse packing trips because some of the springs we sample are so far back, it's better just to camp. And we've had do some day hikes, stay in the middle of a week-long sampling trip. One time we had a bear chase all the other people out of the, well, they evacuated the entire area. It was just us left and the rangers. So the rangers let us take their canoes and go fishing. They were very nice to us. So we do see a lot of bears, but we make a lot of noise while we're in there sampling, and we're not there really to see the wildlife. We're there to see the hot springs. And so most of the people in my group, they like to sing Disney songs for some reason while we're hiking on the trails, really loud to keep the bears away. So most of the bears we see are going the opposite direction. We've never seen a wolf back in the Heart Lake area, but seen wolf tracks. So what are we doing? We've got hot springs that come out at near boiling. 91 C is just 1 degree lower than water boils at that elevation, at 92 degrees C. So nearly boiling, high pH. As the water flows out of this spring, it cools, and you can see this green band here. Photosynthesis can only occur up to 72 degrees centigrade. And so what you find is this very sharp line is essentially the 72 and 73 degree temperature line in this flowing system. And so we've sampled all these different temperatures and pHs to see what organisms are there. We've extracted their DNA, sequenced the DNA to figure out who's living there. And I won't go through all the Latin names because I know nobody's going to remember that tomorrow. But what I want to point out is we've got some that we know the names of. The size of these balls are related to the numbers of organisms in those categories. The balls here in the unknown archaea category, there's more unknown organisms in there than there are known organisms. They're very different. We don't even know what names to put on them. If we could grow them, we could name them ourselves, and so we're working on doing that. To put it in perspective of how unknown these organisms are, if we did the same thing, we wouldn't know that this level of knowledge, we wouldn't know the difference between an orca whale or a puppy. Right? That's sort of the genetic base, the genetic difference of these organisms. It'd be like if there was some creature over there, we wouldn't be able to tell you whether it was a whale or a dog. That's how different these bacteria are. We've managed to grow some of these, we're trying different ways to grow, managed to grow some of these on electricity. Usually you need, we eat things like pizza and we breathe oxygen. And the pizza provides electrons for us. The oxygen helps take what's called electron acceptor. The oxygen takes the electrons out. These organisms can do the same thing, but if we could do this on sheets of carbon fiber electrodes, and they will also grow. So we've tried different ways, taken these electrodes and put them in the hot springs for periods of up to 6 weeks, and then come back and see what organisms have grown on these. What we found is the hot springs, I'm just going to focus on these guys, the hot springs that are very high temperature, above the photosynthetic limit, where there's not much food in the system, responded very well to, this is what they do without electrons, this is what they do when we give them electricity. And so we got over an order of magnitude, 10 times more organisms growing on these electrodes by putting electricity in. So we're looking at ways to grow these, make biological products using solar energy, maybe biological products in space on electricity where you wouldn't bring up those kinds of materials. So we're actually working with NASA on a variety of projects right now, growing biofilms from the International Space Station. But this is one area that we're interested in, using these electrodes, using electricity to grow biomass. We've got a lot of people that are interested in research in Yellowstone. So these are some researchers from NASA's Jet Propulsion Lab that came out and sampled with us. they're working on a DNA extraction sequencing or amplifying and sequencing process. We did the entire process in the back of a Suburban this summer. And so they're working on developing it to put on the International Space Station. So if somebody gets a bacterial infection, they can take like a mouth swab or something, analyze the DNA and get the sequence data right out of a nice little box. Real time. Not quite real time, like all day, one day kind of thing. We go down sample all times of years. These springs, in the winter, we snowmobile about 2 hours in and then hike about a mile in. But it's always a good time sampling. Yellowstone's a great place to work. One of the things we started just recently is an extreme microbiology of Yellowstone undergraduate course. And we've taught it 3 times now. And we basically get undergrads, we only allow 12 students in at a time. The first week we spend doing training, bear safety, backcountry, hot springs training. The second week we spend camping in the backcountry in Yellowstone and sampling these hot springs. And these are freshmen and sophomores. They're new to everything and they just love it. I've got a video. Let's see if I can do this. So essentially, take them in, do some sampling in the hot springs. They collect their own data. This is a sophomore at MSU. Have them do their own pH, temperatures. We always do a day hike in the middle, so we hike up to Mount Sheridan or around the lake or something, and get them interacting with professors and research faculty. We pack all of our gear in on horses, so we don't have to carry everything on our backs. Camp right next to Heart Lake. Again, sharing a campfire with 2 research faculty and a freshman, I think, there. Anyway, it's a fabulous class. We really enjoy doing it and hope to continue doing it through the honors program. That starts over. So far, a lot of this work has been funded by the Keck Foundation. A few years ago, actually 2015, I wrote a proposal and the Keck Foundation to do this work. The Keck Foundation funded it, and on the 2 days after Christmas, an actual $1 million check arrived in the mail. That's how you find out if you got the award or not. You either get a note that says, sorry, next time, or you get $1 million check. I'm pretty excited. So we're using very modern DNA sequencing technologies to figure out how are the organisms in Yellowstone similar or different to thermophiles all over the world. And so we've got samples from China, Tibet, Malaysia, Russia, Japan. I won't even read them all, but all over. These are the ones in Yellowstone. And basically, I'm not going to go through the axes, but they're basically DNA sequence differences. And so the organisms in Yellowstone are genetically different from the ones in Tibet, and genetically different from the ones in Romania. So they have kind of evolved to be their own species or their own genetic code. So we're trying to figure out how similar, how different they are, and based on distances or what really drives those genetic differences. We're also, I mentioned growing organisms on lignin. We're using, this is a electron microscope picture of lignin, so it's kind of a brown, it's the brown stuff that makes trees stay standing up. It's very, very strong. It's very hard to degrade. Well, we found an organism in Yellowstone that you can see the bacteria growing on this lignin surface with these pictures. And we're continuing to work with this to try to get it to break down lignin at high temperatures, and you can make things like a lot of flavor compounds or aromatic compounds that can be made from lignin, like vanillin, a number of different aromatic compounds. We're also looking at using these organisms to break down plastics, and so we've taken PET as the stuff, the plastic in water bottles. So we take a little slice of this PET film, put it into a test tube or a flask with the microorganisms and give them some time. We measure the amount of oxygen that they consume, and the only food in that system is this plastic. And so we're finding organisms that can break down at 70 degrees C in 2 weeks, break down these different plastics. So there's nylon, polycarbonate, polyhydroxybutyrate, which is an artificial plastic, bioplastic. And so we're working on using this data to get more funding to look at using thermophiles to potentially help us recycle a number of very recalcitrant plastics that we have in the world now. I'm going to switch gears just a little bit. Some of the other research that I'm involved in is we started looking for production of biofuels from algae. So algae, turns out, make a lot of oils, and a lot of the oil that we're pumping now probably came from algae millions of years ago. And it turns out that they make oil that's very similar to olive oil, and you can actually buy algae oil in the grocery store now in a product called Thrive. So it's a algae-based oil. They say it's got a higher smoke point, and I can't tell the difference in the taste, but there are some algae products making it out to the world. So how do we screen these organisms to see if they make enough oil to even be worth looking at? It's fairly simple. This is what they look like under a regular light microscope. If you stain them with a stain called Nile red and look at them under a fluorescent microscope, they turn bright yellow if they've got oil in them. And you can even, if you look carefully, you can see the droplets of oil inside those algae bodies. Okay? So we started trying to figure out how do we make them make more oil, how do we make it faster? And so we just going to look at this graph here. This is the yellow, the amount of oil in this system, and this is time in algae. And under normal conditions, it takes about 2 weeks to make enough oil to make it worth harvesting those algae. What we figured out is if you add something right here, there's kind of a critical point when they run out of nitrogen. It turns out if you add baking soda at just the right moment, they will produce the same amount of algae oil in half the amount of time. And so we published a number of things on that, a number of papers on that, but we also got a patent, both nationally and internationally, basically to add baking soda at just the right time to these algae cultures to accelerate their growth. Turns out that this works also on diatoms. These are little photosynthetic organisms that live in glass shells. Diatoms are just beautiful little creatures. Basically, they have little glass shells that they make, silica shells. And it turns out they're sort of the same. These are under light microscopy. Under Nile red, you see they turn just bright yellow. These guys are about 70% oil by weight. I'm glad I'm not 70%. And so some of these make so much lipid, so much oil, that they actually burst their shells open. And they do the same thing. We add sodium bicarbonate at just the right time. This is without sodium bicarb. We double the rate and double the production of oil. So Arm & Hammer like the idea so much they've given us $350,000 to continue this research. And one of the areas that we looked at, so if you think biodiesel, regular diesel at the pump is cheaper than bottled water. And so there's just no money to be made in biodiesel at the moment. But astaxanthin, which is the pink color in flamingos and the pink color in your farm-raised salmon, is $2,500 a kilogram. If we could do something with that, that might be something valuable. So we started looking at this high-value pink, bright red carotenoid called astaxanthin. It is what gives lobsters their reddish color. You can buy it as nutraceuticals. And basically, these organisms grow as a green color, and then when they're stressed, they make this astaxanthin and turn bright red. And it turns out that we could get up to 7 times higher astaxanthin production by adding the bicarbonate. So my master's student just finished her work on that, and we're working on publications and trying to get her a job with an astaxanthin producing company. Another area that I've been working on is growing what's called cyanobacteria, blue-green algae, for biofertilizers. And so these are organisms that can take nitrogen out of the atmosphere, fix it into part of their biomass, and so when you add these to a crop, they provide carbon and nitrogen. They improve the soil properties, decrease erosion. We've grown a number of different crops. We think there's less impact than chemical fertilizer because there's less runoff. We basically have it trapped in a little cell body rather than being able to wash off with the rain. And essentially, these organisms use sunlight to fix nitrogen out of the atmosphere and make amino acids that can be basically put on the crops as fertilizer. Here's some spinach studies that we've done. Basically, the ones with the circles around them have the biofertilizer added. So there's the cyanobacterial biofertilizer at 100%, 50%. Here's urea, and here's our unfertilized control. And this is just spinach leaf area. But we've also got studies out at the Ag Experiment Station with a number of different 12 by 12 plots looking at barley, wheatgrass, and switchgrass as a biofertilizer crop going. So we're going back every year. We're in the third year of that study, going back every year, adding biofertilizer and comparing it to urea as far as productivity of fertilizer. The last area of research that I work in, again, mixing the biology and the engineering. This is up in Fernie, Canada. So this is a company called Tech. Been working with them for about 7 years, and they have a selenium problem. When they pile all this waste rock up, so they mine these pits out. You can see there's a road, there's a haul truck down there. This is a huge operation in Fernie. They pile all this waste rock up, and when it rains and snows, it leaches selenium and nitrate out. And I mentioned selenium and nitrate already tonight, at least once. So we started studies in test tubes, the size of my laser pointer here, proving to this company that the microorganisms could reduce the nitrate to nitrogen gas, so it just goes off into the atmosphere, and precipitate the selenium and immobilize it in the rock. And so we started with test tubes. We went to column studies. We went to pilot tests, and now we're doing a full-scale test treating 2 million gallons a day. We're using methanol as the carbon source, so we're putting a metric ton of methanol in a day to feed these organisms that already live there, to break down the nitrate and precipitate the selenium, and the water is coming out at drinking water standards for about 1.5 years now. This is the water that flows into Lake Koocanusa, so it impacts Montana. We're working with this company to decrease the amount of selenium so that they meet their drinking water standards and fish water quality standards. Essentially, it's just back to biofilms and knowing what organisms are there and getting them to degrade the nitrate and precipitate the selenium. So back to stuff that I was working on when I first got out of school. It's kind of come around and actually being used. The company is working on, Tech is working on 2 or 3 more systems like this, and I can't really say how much money they think they're going to save in the next 5 or 10 years, but it's more than everybody in this room could ever imagine probably. So essentially, this is what the area looks like. These are the pipelines that we're using to pump the water into the waste rock and treat that. It looks kind of like the moon. Eventually, they do kind of cover it with grass and stuff. But essentially, we're working to protect the rivers and the creeks from the existing mine water that's coming out. There's a video, if you want to watch it. It's on YouTube called Elk Valley Water Quality. And essentially, it's based on protecting the Elk Valley. I'm going to go through a few acknowledgments. So this is my research group. Very hardworking young people, great people to work with. A couple of postdocs and students from Spain, Turkey, a variety of places. But anyway, fabulous group of MSU students to work with. A number of folks at the company called Environment. Lisa Kirk was one of my PhD students and spun out a mining environmental remediation company, and now she's got an office in Bozeman and has hired about quite a good business on mine remediation work. All the folks from the Thermal Biology Institute that I work with, and some of our funding from the Turner Foundation, I already mentioned the Keck Foundation, U.S. Department of Energy. MSU supports us pretty well as well. A lot of our algae work is funded again by the Department of Energy and the Department of Defense. I'm not going to read through all the names, it's just too many people. And then our Keck Foundation project that we've got about another year on studying the thermoplastic in these hot springs. So I just want to say TBI just celebrated its 20th anniversary, so we've been around a while. I hope we're around another 20 years. So one of the things, you guys, most of you are alum, so that means you were students. And as a professor, I feel like I should be able to assign some homework. So go to the TBI webpage, type TBI Montana or something, you'll get to it. Root around on our webpage, look at some of the outreach that we do. We've got a program called Extreme Yellowstone Expedition. It's a series of workbooks targeted at middle school teachers and middle school students. It's got student workbooks and manuals, teachers' manuals you can download for free, going through coursework and science curriculum based on Yellowstone. We've got all of our seminars, all of our, we do a book called Living Colors that's sold in the park. Once you've done that, then scroll down to this little blue button here. I'll blow it up for you. Just follow the directions. I want to thank my family. They're very supportive and loving, really outdoors people, and thank you guys for coming.
Okay, back on. Boy, college professors usually don't end a little early, do they?
I'm an engineering professor.
An engineering professor, right, he's quite punctual. So we have time for some questions. Is that okay? If people have some questions they'd like to ask, yes, sir. We've got a mic coming to you right behind you.
Oh, I don't need a mic. What is the role of oil in the life of algae?
So algae make oil the same reason that we make fats, right? When in good times, we eat too much pizza, we drink too much beer, we accumulate lipids. They basically do the same thing. If they get a lot of sunlight, they have a lot of carbon dioxide that they can fix, and they keep growing. If they can't reproduce quite when they want to, then they store all that energy as lipids, and they can respire it with oxygen. They can use it the same way that we use oils.
Go ahead.
I'm curious, do you have any organisms or anything that can solve the Berkeley pit problem?
I
Berkeley
wonder where
pits.
she got that question.
Yeah.
Okay, go ahead.
So, years ago, we looked at the Berkeley pit problem, and the sulfate-reducing organisms that I talked about that could precipitate the copper and the cadmium and whatnot, that's a lot of the problem in the Berkeley pit. We looked at how much food it would take to feed them to treat the amount of material that would be needed. If we took all of the biggest source of food that we could find was the Butte sewage treatment plant. If we took all the sewage in Butte and converted it and used it for bacteria food, it still wouldn't be enough food to clean up the Berkeley pit water. It would do it. You could precipitate the metals, essentially you put the sulfide in and precipitate the metals, but there's not enough. You'd have to still bring in truckloads of other carbon source to feed those bacteria to treat that was the problem. But yeah, very good. I mean, it's certainly possible. There's a question back here.
Question.
Raise your
hand, please, back there, whoever had a question. Oh,
Maybe she doesn't want to.
okay, questions, yes.
I would like to know if drones are being used in Yellowstone for some of the collection when it's not possible for humans to collect it.
That's a good question. Yellowstone just banned drones from like everyday use. I think you can still get a permit to use a drone, but we get asked that a lot. And there's so many springs that we can just get to with we get those second story window washing poles, right? So it's long and they put a big sampling cup on it. We can get to most of the stuff that we need to get to with those, and there's another 10,000 springs that have never been sampled. So we haven't had to resort to drones yet. And the park doesn't really like them because if they crash and they're in the hot springs.
Okay, other questions?
So you mentioned the SRV thing,
Mhm.
and you talked about precipitating the copper and stuff. Does that mean that we can then utilize the copper to make copper things or nickel things?
It would be in the form of copper sulfide, and you could collect it and send it back to be smelted if you were doing it above ground. If you're doing it underground, it would stay there. So yes, if you were doing it in large tanks above ground, you could collect the precipitation.
And then when you're doing samples, are you just dipping in the water? Are you scraping the edges?
In the hot
How do you
springs?
get your samples? Yeah.
So sometimes we sample the water, and sometimes we have a stainless steel cup on the end of our sampling poles, and sometimes we take scoops into the sediment in the hot springs. We usually do both actually, and see significant differences in the two. Sure.
A couple of years ago, one of your cohorts was here and talked about a new paint, perhaps from the algae in Yellowstone, that would make ships slide through the ocean easier.
Okay.
Do you know if that research has progressed, or?
I haven't heard that it has progressed. I'm also in the Center for Biofilm Engineering, and that's what we do, a lot of stuff to try to keep biofilms from growing on things like ships, and I haven't heard anything about that in years.
I wondered about the amount of basic research as compared to commercial.
At MSU or me? At MSU. Most of the research I would say at MSU is basic research, but a lot of it has potential applications. It's like when we're trying to understand what organisms are in the hot springs, a lot of that is just very basic and will never result in anything commercial. But some of the enzymes that could maybe be used to break down cellulose, to make sugars, to make ethanol, could be commercialized. And so it's, I would say, mostly fundamental, but MSU has quite a few patents every year as well. Okay.
Montana has a deep history of cyanide heap leach. Are there any organisms that will digest that stuff?
Yeah, I'll repeat. I have a microphone now. So the question is, Montana has a strong history of cyanide heap leaching used in gold. And there are organisms that will break down cyanide, but I don't know of any organisms that could replace the cyanide leaching that would be as efficient at getting the gold out of the ores as the cyanide is. But bacteria could be used to break down some of the cyanide contaminants.
So speaking of other applications, what about the microbes that live in the soil? Have you looked at, I know you're focused mainly in the hot springs, but what about like other contaminated soil? And I was wondering if like the EPA has been interested in any of your work.
The EPA has worked in parts of these areas for a number of years, and so they are, right now, the Canadian equivalent of our EPA is very interested in the work that we're doing out in Canada, and other companies are starting to copy that treatment process, and so, but I mean, we could go out into the soil outside this building and find organisms that would precipitate silicon. We would just put them in a beaker and grow them up, and eventually we'd have some that do just fine at that process. So it could be applied just about anywhere.
Have you done any comparisons of these surface extremophiles with the deep sea vents?
Good question. We do, sometimes when we do DNA extractions, we see some similar organisms in our hot springs to the deep sea vents, but that's about as much of a comparison as we do currently. We try to identify the organisms and then identify what enzymes they may have that might be potentially useful. I'm actually working on a proposal right now, it's actually due February 6th, that is for a $3 million project to study biofilms in extreme environments and teach. We'd have about 15 PhD, if we get it funded, 15 PhD students studying those kinds of things. So everybody cross your fingers.
Do you give tours year round?
Tours year round. I have, yes. If people want to organize something like a class tour or alumni group tour, we're just, as long as we get it on my calendar far enough in advance.
And we asked that question earlier, a small group of us, and I think you contact the alumni office, and they would put together the list for you.
Tell them you really want it, and they can find us.
That's the people we talk to.
So, in your study of biofilms, how important is the community structure? So you might have diversity, or do you focus on just identifying and extracting and working with a single population?
So a lot of the work historically has been on as monoculture or single species. What we're trying to do with the DNA sequencing, we can separate out the different species. We have ways to actually develop fluorescent probes that can tell us where each species is living and separate 15 different species. And so we're doing more and more and more with mixed cultures, and also mixed cultures of bacteria and algae, bacteria and amoeba. So we've got a project with a group in Australia looking at the Eugleria fowleri, the brain-eating amoeba that people get once in a while. It actually eats some of the high-temperature organisms that show up in some of the Australian drinking water pipelines. And so we've working to identify Eugleria and what organisms they live on here. And so definitely mixed cultures are much more real in the world, and we're doing as much as we can to understand them.
Dr. Payton, you obviously have been in this research area for many, many years, and you have made many discoveries. Of all the discoveries, what has been the most thrilling for you, and what's the future for you as far as a major breakthrough?
Oh, geez. So I think over the years, there's been probably two times that I felt like the discovery that we made was ahead of everybody in the world. I mean, big time, not just little things. One of those experiments we didn't believe, so we ran it like 7 times with multiple controls and as many controls as we could. And anyway, is when we presented those results, it was dead silence in the room, and people couldn't believe the results. Later, it was confirmed that it was true, but it was so a couple of times. As far as future stuff, I don't know. So our baking soda work for the doubling the production of biofuel. We need a couple more doublings, so if I could find something that would do that 2 more times, that would be fabulous, and actually get biodiesel down into the range that we could make it and afford to buy it. Bring it down in the $2.50 a gallon range instead of $15 a gallon.
Yeah, that's good. How many students do you have in the chemistry department, in your grad school, and in your post-master's program?
Oh my gosh.
Roughly.
Chemistry department, we probably have about 500 students now, up from, I remember telling the dean, I was interim department head 1 year. It's the worst year of my life. Yeah, actually. And I remember telling, we had about 350 students, and I remember telling him, I think we're going to be above 400 next year. And he's like, no way, you guys never. And we were up to 600 a couple of years ago, and it's actually tapering off a little bit, which is good. Grad student numbers, you know, I don't keep track of how many at MSU. We have about 25 to 30 in the department.
And in your undergraduate program, how many are in that class you're teaching?
In the undergraduate honors course?
Yes.
12. We do 12 a year. We're talking, so we do that one in the fall. We're talking about trying to expand it to a winter one where we would, instead of hiking in, we'd snowmobile in and snowshoe in to sample. And so I think that one could be just as interesting.
Okay, one more question. Anybody got another question here? Yes, back there, Bill.
What's your feeling about the prospects of dealing with our plastic problem?
Plastic, so what do I think about dealing with our plastic problem? I think plastics, I mean, some of the, we were down in sabbatical in Australia, and some of the islands around there are just inundated with plastics, and we have Texas-sized rafts of plastics in the Pacific. Plastics, some of the microorganisms are starting to figure out how to eat plastics, very, very slowly. Yeah, they're figuring it out for themselves, right? There's a lot of food out there. If they just generate the right enzymes, they can break it down the same way they do cellulose or lignin. We're working on proposals right now to try to come up with enzymes that can break plastics down into their primary components so that you could recycle like a virgin material. I think that's a really nice way to go. I think the other thing that we ought to all do is drink less bottled water, everybody, please. And if the U.S. would go to more biodegradable plastics. There's a lot of plastics out there. They're a little more expensive, so you pay a nickel for your plastic fork instead of a penny. But I think we need to go to biodegradable plastics and figure out how to recycle the ones that we have.
Okay, Brandon. Okay, well, thank you. Before we get up, if you're a teacher, Ann, would you stand up again? Ann Simpkins needs to talk to you, or make sure she's got you on her list. Okay, 2 weeks from tonight.